I would like to mention that Dr. Tara Satyavathi, she is the coordinator, project coordinator for All India Coordinated Research Project on Pearl Millet Improvement of Indian Council of Agriculture Research based in Jodhpur, India. And since she has taken the reins of this AICRP-Pearl Millet, the Pearl Millet program in India is reaching to the new heights, new levels. And in one of the important area that after she became the project coordinator, she has introduced a concept of minimum iron and the zinc content in the promotion criteria and varietal release policy in the case of Pearl Millet. And this is the first time ever in the world. And as a result, when she has led these things, they have been successful to release about 15 hybrids and varieties with minimum 42 ppm iron and 32 ppm zinc, leading to the nutritional security of rural women and children. So this is one of the important topic of our webinar as well, that basically we are talking about the genomics, not only for the food, but also nutrition and health, and iron and zinc, they are important micronutrient, and we are very happy to mention that our co-chair is expert in this area. And under her leadership, research efforts addressing other traits also related to the consumer, like rancidity in Pearl Millet, and also development of the Pearl Millet material in that arid zones in India. And this arid zones indicate me that issue of the drought, and this is the region that we also have one speaker talking about the drought tolerance as well. Dr. Tara has received many awards, including Dr. Punjab Rao Deshmukh Outstanding Women Scientist Award, Outstanding Millet Scientist Award, Outstanding Research Contribution in Pearl Millet Improvement in the country, and also MS Swaminathan Women Scientist Award. And as you can see that in this session, we have all women, co-chair, we have chairwoman and all the two speakers women, which also bring very important aspect of our webinar today. And she has also published several papers and received many awards. And one of the other important thing is that she is very good collaborator and great friend of ours. So we would like to welcome you, Tara, for this chairing this session. Now I hand over this mic to you, and you please chair the session. Thank you, Tara.
Okay, thank you, Dr. Rajeev. And I welcome you all to the second session of this webinar on genomics for food, health, and nutrition. As mentioned by Dr. Rajeev, we have heard about the two good talks in the morning by Dr. Lee Hickey on speed breeding, and then Dr. Bean on heterosis breeding. Moving from these two who are trying to go for the food security of the world, we are moving towards phenotyping. Phenotyping for biotic stresses, abiotic stresses, and also breeding for nutrition. Just before introducing the speakers, I would like to share a few words regarding this. As students of plant breeding, when we joined the plant breeding courses, we were taught what the two principles of breeding. First is creation of variation, and then selection of variation. Selection of variation means how do you go, how you select in the field or in the lab. There comes this principle of phenotyping. If we see how our old breeders have started phenotyping or selection, you remember that all our teachers, our senior breeders, they used to go to field. They used to have a diary, a pencil, a scale, and all those things, carry them, stand in the scorching sun, take the observations, and then bring back the book and then see the data, then process it and analyze it, and then come out with the results. Now, as we move on, the concept of selection phenotyping has changed, and we are seeing a lot of change due to the technological advancements in various fields. And today, we hear not only the term genomics, we also hear about phenomics, metabolomics. Then in one of the talks today, we are going to hear about nutritional phenomics. So the world is changing, the technology is changing, the science is changing, and there are many new additions to the field of phenomics. And you also know that the phenotype is the resultant of the combination of genotype with environment and genetic and environment interaction. So to capture the changes imposed by all these factors, the genotype, the environment, genotype into environment interactions, we have to have very fine tuned equipment or tools which can capture these changes and help the breeders to select those things. That is where the phenomics come into picture. And if you see the phenomics, it is just not a single discipline. It is an integration of many basic disciplines involving mathematics, physics, chemistry, computer science, computational biology, and biometrics, and electronics, many things, along with artificial intelligence. So friends, today we have two eminent researchers in the respective fields who are going to give talk on different topics. First, we have Dr. Kerstin Neumann, then followed by Dr. Dil Tavaraja. First, I'm going to introduce Dr. Kerstin Neumann. Dr. Kerstin Neumann is a long-term scientist working in the Leibniz Institute of Plant Genetics and Plant Research, popularly known as IPK in Germany. She studied biology at the University of Potsdam and then conducted her PhD work at the IPK in the group of resource genetics and reproduction, working with a diverse barley collection on mapping drought tolerance at different developmental stages, applying genome-wide association scans. After her PhD, she started working in the field of non-invasive high-throughput phenotyping, that is HTP, in the group genome diversity. Since then, she employed HTP in different panels of wheat and barley for mapping abiotic stress tolerance related traits. HTP in controlled conditions allows detailed insights into the dynamics of genetic architecture of complex growth and stress tolerance related traits. Dynamic trait mapping has the potential to enable breeders to include time-dependent QTL information in their processes. Dr. Kerstin Neumann is a member of the German Plant Breeding Association. Dr. Kerstin, the floor is yours.
So now I have to put my mic on. Is everyone listening?
Yeah, we
good. That's the first challenge. So thanks a lot for inviting me to the seminar. And the webinar is actually my first webinar and the first webinar given at IPK. So we are looking and observing if that works fine. It might be also a platform for here. And yeah, I have to share the screen now. So presentation is here.
And Kerstin, you already got around 1,500 participants on that together on both platform, and very soon number of participants will be increasing further.
Okay. So I just have to say, I have to line out this one. All right. So I hope that you can see my slides now. I'm in the presentation mode now.
Before that, can you put on that slide full view? Or Prasad, just help?
Yeah, that's what thing. Kerstin, if you can go above over there, there's an option of display settings.
Yeah. Wait, that was yesterday the same. With this swap, yeah?
You can just quick swap, or you can do duplicate slideshow as well.
This is okay, good.
This is good.
Is okay. All right, so first technical difficulties. Yeah, so the seminar or the talk that I'm giving about is probably on the brink of applied and basic research. As an Institute of Plant Genetics, we are interested in the genetic architecture of traits. And yeah, this is my work since a long time, to work on complex traits correlated with drought tolerance or abiotic stress tolerance in general. And we employ the diverse mapping panels for that, yeah, which mainly accessions coming from the gene bank, although not only. And the seminar I want to give today will focus on results coming from this evaluation in barley. And as you see here already on the picture, we are employing non-invasive phenotyping. Also, we do invasive phenotyping, but the main talk is about the non-invasive phenotyping aspects and what you can learn from these studies and what traits you can extract from these studies which are relevant for drought tolerance. And before we go into details of that, I thought it would be maybe a good idea to focus a bit on the ongoing climate change under German conditions. So are heat and drought of any relevance for Germany? Because maybe from India, if you look on Germany, you would think, well, this is temperate climate. We don't have an issue with these stresses, but that's not fully true. And particularly, you will see not for the last 2 years. So while the overall annual precipitation has not changed in Germany since the climate change and is not predicted to change in the total amount of precipitation, the prediction by climatologists are that you will have a shift in the pattern of rainfalls with more winter rainfalls and less summer rainfalls, particularly in the northeastern region of Germany, which has already very light soils. Is a prediction that up to 30% of the rainfall in the summer will decrease. And there are some new studies which even suggest that this earlier prediction that we have a higher amount of winter rainfall that could compensate for that would not be fully true, but we need to see how this develops. So let's say the annual precipitation is not really touched by climate change. However, the summer rainfall can be reduced. And of course, as every country, we experience increase in the temperatures. As you see here, this graph is the example of Gatersleben. So we have our own weather station since 1953, and these are the annual mean temperatures for every year. And you see clearly a significant increase also at this location with an R square of 0.34. And I have highlighted in yellow here years that have higher temperature than 10 degree in average. And you see they all occur in the, since the 90s. They didn't occur in the 50s, 60s, 70s, 80s. And this is also increasing. So in the last years, we even had 3 years which were above 10.5 degree in the annual mean temperature, which includes all the winter temperatures and so on. Of course, it doesn't give you really an overview of is heat going on in Germany. We looked a bit more closer at the data in the weather database on the daily temperatures. So since the year 2000, you have this information in the database, and I extracted the amount of days that is higher than 30 degree. Which is mainly in the summer, of course, but can also nowadays occur already in spring. And also in this last 20 years, these days have significantly increased. And I just want to highlight here, there's a year 2003 with 27, let's say, heat days, which was a heat wave across all of Europe, known as a summer of the century, which caused also death, heat death, people dying on the too warm temperatures in the towns, especially old people. And this heat summer could be directly attributed to climate change by climatologists. However, that is nothing compared to the last 2 years. So in 2018 and 2019, we had even much stronger heat waves, especially in 2019 with 41 such days, but also 2019 had 31 of these heat days. And you see this is not only true for Gatersleben, this was true for all across Germany. So you see here the map of Germany, and this is from the German Weather Service, DWD, that compared the deviation of the summer temperatures in 2018 with the long-term average of the normal period from 1981 to the 2010. And we can argue if that would be the normal period, because already this period is warmer than the 50s and 60s. But even then, you see a strong increase of the temperatures in 2018 from plus 0.9 degree in the whiter areas to the reddish, the more red, up to more than 3.8 degree. So it was the hottest year, or the warmest year ever recorded. Since the beginning of the weather records in Germany, which started 1881. Moreover, 2018 was also the driest year since Germany started the Drought Monitor, and this is basically what you see here on the left side. So this Drought Monitor uses data from soil moisture in combination with the drought period length and also the geographical location. And this is monitored from April till October for every year. And you see there are years, and you see here the 2 years, 2018 and 2019. So there was a stronger drought period in the early 70s, from starting in 71 to develop, and then 76, the strongest year. But this is nothing compared to the past 2 years in Germany. So they also state that 2018 has been the driest year ever recorded in this drought monitor. And 2019 was a bit, and this was all across Germany, while 2019 was a little bit different. So we, in the regions that were hit the second year of a drought with not enough rainfalls, this drought was even stronger than 2018. But there were also regions that had some more rainfalls in 2019 and where the drought was not so pronounced. However, in the regions, and this is pretty much the pattern they were predicted by climate change, in the northern eastern region, also the southern region, you had a second year of drought, which was even worse because you already started with a water deficit in the soil. And in 2018, 70% of all of Germany's total area suffered from severe water deficit and also caused strong yield reductions. And 2019, we had a bit more rainfall, but as said, the water deficit was stronger in the soil already. And we will just look a little bit more in detail at the Gatersleben location. So you have to keep in mind the average annual precipitation of IPK or Gatersleben is about 570 millimeter. And in 2018, it was in total only raining 263, while in 2019, it was a bit more with 385, but that's still far below our normal rainfalls. And you see here the monthly precipitation, and you also see that especially here, September, October, November, December, basically the whole winter period, where usually the winter rain should fill up a deficit that has maybe developed over summer, was also very poor and couldn't fill the deficit. So we went into a drought again in 2019. Here in green, you have the growing period of the cereals. So that's from March to July. And this is here a bit more resolved. So that's the daily precipitation. So starting with the 1st of May and ending 31st of July. And while in 2018, which was a year where the drought developed, we still had some rain in the spring. It was almost completely dry in May and in June, and also really little amount of rainfall in July. And that in combination with very high temperatures during that period, which then caused the drought towards the grain filling period to be really bad. As said, the winter rainfalls couldn't compensate. And moreover, here in Gatersleben, we had almost no rain in the early growth stage period. So in March and in April, there was almost no rain at all. And it also rained only very poorly during the summer. So basically here, the 2019 year was far worse than 2018. And just as a comparison with the total of Germany, so there were data comparing the rainfall from April to 30th of September 2018, which was all over Germany drought. And they had an average 263 millimeter across Germany, while Gatersleben had only 148 millimeter rainfall in this period. So we were even hit worse than average Germany, and 2019 even more. Yeah, as said, 2018 has caused also crop failures sometimes even, and reductions in the cereal yield, which are reflected here in this graph. These are from the Statistica website and starts in 1916, where of course breeding has not yet resulted in such high yields, but then the yields increase. Here's 2010, and then you have every year to 2011 until 2019 in this graph, and you have here the barley, and you have triticale and rye. And you see that since 2014, the yields go a bit down, and the worst harvest we had in 2018 across all of Germany, while in 2019 in the German average it was a bit higher because not all regions experienced that second year of drought, but in the regions where they were hit with the second year, it was even sometimes complete crop failure. But in the German average, it was still better than 2018. But that also gives you a hint that here breeding also needs to shift to more drought and heat tolerance, because also that came along with heat waves. And it is clearly an issue. And we even had or have the fear that we enter a third year of drought, because also the precipitation in 2020 in this spring has been, let's say, absent, because there was barely any rain since middle of March until the whole April. Here it rained a little bit, but I mean 4 millimeters is still not enough. And just the last days it started raining, and we hope that this will last. But what we also saw that in this period we again had higher temperatures, so the crops also developed faster. So the rapeseed flowering is already over, which is very unusual, while winter barley already is also in the flowering stage, and that might also cause crop yields already. And you see here that the Drought Monitor on the 21st of April 2020 again tells you strong inhomogeneity of Germany, with the southern region and the eastern region hit by drought again. And this can be only compensated if it's really raining a lot, because we almost have a full amount of annual precipitation missing here, and this soil deficit has to fill up again. But we hope that it's continuing to rain over a long period of time now. Just to give you some illustration, so this is basically how the maize looked like here in this region that usually has a very good soil and a high yield last year. So I never seen wilted maize in our region at all. And last year it was almost a complete failure. Also the soil was very dry. This is an image from northern Germany. And there are very bad predictions. So if the global warming cannot be kept at 2 degrees, but increases to 3 degrees, that a lot of federal states will be hit by drought periods, or by an increased amount of drought periods. So Germany may more often look like this due to the ongoing climate change. So it is an issue also here for breeding to develop cultivars which are resistant or at least adapted. What we do is we try to evaluate a lot of traits non-invasively, but also invasively, which are all affected by the drought, because drought affects plant growth at any stage. So the root system plays a role, of course, the root biomass, the depth, the number, the angle, and so on. Drought can reduce the biomass, the tillering, can affect the early growth vigor. It has an effect on the phenology that drought can cause earlier or later heading, depending when it hits the plant and how strong it is. It can, of course, let plants get earlier mature. And it has, of course, an influence on all the yield components. So what we are trying to observe is the above ground growth of the plants using this non-invasive phenotyping methods. And we also have a look at roots with different methods, whether destructive or non-destructive. So it can be these simple screens and filter paper, cigar rolls, or you have here hydroponic platform now, or you can also do shovelomics in the field. And all of this we want to combine then with genomic information that we have to, in the end, identify genomic regions that are linked with drought tolerance traits, and of course explore these regions for potential candidate genes, and in the long term and improve the drought tolerance or contribute to improve the drought tolerance of crops here. You already mentioned phenotyping. So phenomics has become a second big issue here in IPK. I mean, we are a genomic center, but without the phenotypic information that you can link to this genetic information, you also don't see the full picture. So IPK has become one of the centers of phenotyping as well. We're part of many phenotyping networks, and you can also look on the infrastructure here on this link if you're interested. So it would be a complete own talk just to talk about the phenotypic infrastructure that we have. Just to highlight, we have 3 of these platforms from a company named Lemnatech that does imaging and watering, and we are using one of the systems for exploring the drought tolerance. And the latest baby, let's say, is the plant cultivation hall, which is a worldwide unique facility. It's basically a big climate chamber where you're completely independent from the environment, and you can conduct experiments in field-like but controlled conditions. So you can repeat the conditions in the field. So you can simulate from light conditions from winter until summer, and you can also model, let's say, the temperatures from a winter period to an extreme hot summer in this hall. And in one compartment you have the rhizotron system, which was installed just half a year ago and is tested at the moment. And in the other half you have a container-based system where cameras will evaluate traits from the top view, and these are then more like mini plant stands. While in this pot experiments here, you have mainly single plant growth. So what we are using is one of these Lemnatech facilities, the oldest one in the public domain. So it was kind of the prototype. And so we use that since long time for cereal growth. It has in total 520 plants, which you can evaluate on the system. And they are standing here in pots on these carriers and can be transported with these conveyor belts to the imaging chambers. And you have different imaging system from visible, near infrared, fluorescence. Nowadays also quantitative chlorophyll fluorescence installed since 2017. And you have a top view camera and a side view camera for these phenotyping or camera systems. And more importantly, you can conduct controlled watering, which is very important for drought experiments. So you have a balance pump station that is connected with a water tank, and you can water the plants after the imaging period, each carrier individually based on a target weight that you calibrated before to a certain water status, or you can also give a total amount of water. So, and all these data are recorded and then have to be analyzed and exploited for the traits of interest. So, of course, you cannot work with the images, so you need an image analysis. And this, are 2 example images from a side view of the visible light and the top view of the visible light. And you can load this experiment into this integrative analysis platform, which was developed by the former head of the image analysis group in IPK, Christian Klukas, who was here a long time and set up all the image analysis. And he has developed a really sophisticated system also for analyzing these huge data sets. And mainly what the algorithms have to do is to extract from these raw input images what is really planned. So it has to calculate background versus foreground and leave only the plant pixels. There are pipelines for different crops and the different systems. And you see this is one of the result images that works pretty well. So this is what is left after the image analysis. And then based on this, you can calculate a lot of features, of course, from all the 3 different camera systems. And you end up with a lot, hundreds of phenotypic traits, which you have over time and which you can subject to phenotypic and genetic analysis if you want to. Let's say there's some main traits that we can easier grab that is like a trait called digital biomass, which is basically a volume, a pixel volume, which is correlating always excellent with when you measure the biomass in terms of fresh weight or dry weight. You of course get the plant height, you get the plant width, you get something like the compactness, and you get color information which can matter under stress. So each pixel has of course a color value in this color space. Usually it's if it's a healthy plant, it's in the green area. So you can work with the average color value of the total plant pixels, or you can create ratios like how many yellow pixels compared to green you have. That's then the yellow to green ratio. And all these traits can be used for the genetic analysis for GWAS or looking on correlations. What is not yet possible is to count the number of tillers from the images. We do that by hand if we're interested in it. And the same goes for the flowering time, but you can inspect these images visually. So you end up with a challenging amount of data. This is just one example where Dijun Shen tried to present what is possible with all this data. So you have each of these rows here is one phenotypic trait from the different camera systems, and you can look on the genotypic effect for each trait. You can look when you start the drought here at this blue line, when does the trait show a treatment effect? So when does it change drought compared to control, or a G/E effect? And let's say for genetic analysis, it's very challenging to work with all these traits, especially sometimes you have traits that you don't really understand. So we usually use a few of these traits for GWAS. We do, since a long time, use this precision phenotyping platform in different projects. So we have started with barley, and this talk will be about this collection. And we had to establish the setup, did that first with a barley core set of just 18 accessions, and then applied this setup to a diverse collection that we use then for GWAS and QTL mapping. We have then moved from spring barley to winter wheat in the Brivex project, where the data are also ready and need to be published now. Also to spring wheat in 1 year of the Wilby project. And we moved also to heat stress now. And in between, we also had some collaboration in the EPPN project with Swedish collaborators who were interested in scoring early growth vigor in the Nordic spring barley collection. So there's a lot of data sets, but it's impossible to show them all. So we will focus on this data set of the barley collection. As you know, IPK is also a gene bank. So all and the whole barley collection was single seed descended in the Bridge project and subjected to fingerprinting by GBS, genotyping by sequencing. And what you see here is a principal component, so the clustering of this almost 20,000 domesticated barleys, and they cluster based on origins and also row type. And long before that project in our group was established a mapping panel from 224 diverse spring barley accessions, which were already part of a core collection created in 2003, so long before they were fingerprinted. And this panel comprises 224 diverse spring barley accessions, which are since then used in the research community for very different traits, from agronomic traits to salt tolerance, to developmental stages, plant height tillering, and also roots. And it's still pretty much comprising a lot of the genetic diversity of the total gene bank collection when you compare now where this 224 accession cluster on the complete plot of the domesticated barley. For convenience, we selected only a two-row subset of this diverse collection, which is here then in red. So these genotypes went on the LamnaTech platform to be evaluated for abiotic stress tolerance, still also showing some range of flowering time, because also for photoperiod, this set differs a lot. And they were evaluated with this setup. So we have looked on pre-anthesis drought tolerance. We are doing the sawing on the platform, and then having a kind of establishment phase or pre-stress phase. And we started the drought stress in the tillering phase. And it lasted about 3 weeks, so 18 days in special, and or to be precise. And then we ended the drought phase and wanted to look also on the recovery of the plants, because that's here more our conditions, that we have a drought phase and then it rains again and the drought is ended. And you can assess all of that non-invasively, which would not be possible by destructive phenotyping. While of course in the control treatment, you don't subject the plants to any stress. And the experiments ended at about the flowering stage, depending on the genotype, of course. So we do daily imaging and watering, and we have an image configuration that does one top view image and 3 side view images, because you can also rotate the pots and you have images from different angles. We randomize the plants by the system, so we overcome the greenhouse inhomogeneities, and we perform 3 experiments for each treatment, and with 5 replicates or 5 plants per genotype. And yeah, then you end up with individual data. So of all plants, you can plot the growth curves. So this is now one plant from the well water treatment, and you can, with this daily biomass assessment, you can follow the growth over time. And not surprisingly, it follows a logistic model. And from this logistic model, you can extract a trait named the inflection point, which is the point of maximum growth. So before the plant grows faster and faster, here's a maximum growth, and then growth is slowing down and you reach this plateau. This was surprisingly very heritable, so it's about 0.8, and also showed a high range of 9 days in the barley collection, which reflects the range of flowering time, which were found in the field. And we found 2 QTL for this inflection point. So one on 7H and at 34 centimorgen. And this I-select marker was located on the same contig as a well-known flowering time gene, HVFD1. So that makes sense. And another one at 1H, there was no known flowering time candidate gene. And we also haven't found reports on heading time QTL at that time. However, very interestingly, last year came out a publication that published a new QTL for heading time found in a barley magic population, and they named that HV heading, and that resides pretty much at the position of this inflection point QTL. And moreover, several of these magic parents are part of our 2-row GWAS panel that we found here, this inflection point QTL. So that's probably the same QTL, and it proves that this QTL is not an ARFLET artifact, but actually has something to do with heading in barley, and there must be another flowering time gene, but it's still unknown which one that is. Of course, we're more interested on the drought here. So in drought conditions, you have another growth pattern. This is now again one individual plant, and we did this modeling with all plants from all experiments. And here you have more this bell-shaped curve. So here the drought starts at this first dotted line. The plant, of course, still can grow for a while, but the stress gets stronger, and at a certain point, the growth is stopping, and this digital biomass is decreasing, which is reflecting a loss of water. So the leaves start hanging, and you don't see anymore the full area of that plant. And therefore you have a loss in this biomass, and this is wilting of the plants. Once you rewater the plants, which happens here, the biomass is recovering fast, because then the leaves get erect again, and you have a more or less linear regrowth, because here we ended. It will switch to logistic, but we couldn't see that anymore because the plants left the platform here. So what we were interested in to know this time point, when does this wilting start? And this is basically with a bell-shaped model, you can get this time point, which we call wilting time or time A. You also can then assess the biomass at that time. And you can cross that information with the watering information, which you have. And basically we found out we wanted to know at what water status in the pot is that time A for each plant. And that is in average at 21% available water content. Also, this trait had a high heritability, and you see there is a certain range also for this trait in the barley cultivars. So some will only wilt at a very low water content, while some will already wilt at a much higher water content. Also, the time A had an excellent heritability for such a noisy trait, let's say, and also quite a huge range of 6 days in this collection. Later we found, when we repeated this experiment in 2017 with chlorophyll fluorescence, that around this wilting time you also have the photosynthesis decreasing. So here you have the control treatment, and here you have the drought stress photosynthesis. And around the wilting time, you have a decrease of the photosynthesis. Then in the strong drought phase, it's decreasing strongly. But once you rewater the plants, if they are not severely damaged, also the photosynthetic efficiency recovers very quickly. And interestingly, the drought adaptive biomass QTL that we found were in this, let's say, severe drought stress phase. But we will come to that very soon. First, I want to just also present the correlation of biomass in both treatments. Of course, first, it's highly correlated. Once here's the drought is starting, here's this red line. And then this correlation of biomass in both treatments with a severe drought goes down and disappears and goes up again, mainly after rewatering. So you have here in this phase of the severe drought phase, no correlation of biomass anymore of both treatments. That means the ranking of the genotypes is different and you expect different QTL when you compare the treatments. Also the tillering, of course, is affected. So after the drought ended, which is here day 45, so that's the last day of drought, we counted the tiller, and the tillering is severely reduced. But barley has this retillering ability, and in the end, after 2 weeks of recovery, these plants even had a bit more tiller than the control treatment. However, this will be mainly unproductive tiller, so it doesn't mean that the yield will be higher or better. Also, the flowering time has an influence on biomass and implications on drought tolerance, and the panel differs for flowering time. So what you see in this graph is the correlation of flowering time assessed from the images to the biomass over time. And in blue, the biomass over time of control conditions, and in orange from the drought stress experiments. Just remember, the drought stress is only here between these dotted lines. So early biomass is negatively correlated with the flowering time. So the early flowering genotypes have a higher early biomass. That makes sense because they have to be faster with building up biomass. But then this correlation changes and you have an ever increasing correlation. That means the later flowering genotypes will end up with more biomass, which also makes sense. In the drought stress phase, that's of course then interrupted by the drought. And the longer the drought goes, the more this correlation drops and turns even into negative signs. And that means here the later flowering genotypes are disfavored. But of course, when you rewater and the plants recover, you have an even higher correlation to the flowering. So that means the later flowering genotypes can recover also better because they are not yet reaching the flowering stage. What you see here is other parameters like the tiller number from the stress treatment, and here the time A, the wilting time, and the biomass at wilting time. So they are not or barely correlated with flowering time. So on these parameters, the flowering time has no influence. But also flowering time per se is important for the drought adaptation, is well known as an escape mechanism of drought. The time A, however, itself is depending strongly on the biomass at the onset of start. So this is here, this wilting time against the biomass at the drought start in the spring barley panel. And you see there's a strong negative correlation. That means the ones that have at the start of drought a higher biomass will also wilt earlier. So if you want to select now drought tolerant or sensitive contrasting genotypes, you have to look along the same biomass range to really be independent from that effect. And you see here, for example, at a low biomass range, there's one cultivar that wilts very, very late compared to other genotypes with the same biomass at the start of stress. So they definitely contrast for drought tolerance. And here at a higher biomass range, you have another example. And we looked a bit more closer, what genotypes are these? And it's very interesting that this genotype, for example, is a Syrian landrace, so from a country experiencing drought. And this is genetically also very close to Ata and Tadmor, which are two drought-tolerant cultivars developed by ICAR. And the, let's say, more sensitive genotypes of that biomass range are cultivars from Great Britain, Sweden, and the Netherlands. And let's say this most contrasting couple has a difference of 4 days in their wilting time, although they have the same starting biomass. So they differ definitely for drought tolerance. And the other example is here, and there it's again a landrace from Kyrgyzstan that is more tolerant, and cultivar from Canada and a landrace from Russia, which are here more sensitive. And similar, we've seen that in the wheat panel of Brewex, which is a Central European germplasm, elite germplasm. And although this correlation is not that strong, you also have this effect, and you also can identify contrasting cultivars here. Just one example. So these are two German varieties, Rector and Jos, which look pretty comparable in the well-watered treatment, but yeah, look very different at the last day of the drought, with Jos still looking far better than Rector. And this is exactly here. Jos has a much later wilting time than Rector does. So also on this panel, you detect a range of about 6 days in this wheat panel. But here it's far more, it's 220 accessions, and in barley we investigated 100. And in barley, we of course then already have the results for the QTL mapping, and we found 4 QTL for this wilting time. But because of this connection with biomass, 2 were co-located with biomass QTL, so are basically biomass QTL, while 2 were also independent. And we have, yeah, let's say, discovered or in this region of these 2 independent QTL are also genes known to be involved in the drought tolerance. You can then, of course, explore the biomass over time for QTL mapping, and you identify QTL which come up at different times. So this is again the time axis, this is the log P value, and each of the line represents 1 QTL. And you find 3 early biomass QTL that disappear very fast in the well-water treatment, and you have other QTL which come up over time and are more for the late biomass, while this yellow, this violet line here is the only QTL which were more coming up in the middle of the growth period and then disappearing towards the end. So in total, we found here 7 QTL, while in the drought setup, we looked separately here at the drought stress phase. So in that phase, a lot of QTLs come up in this late drought stress phase. In total, we found 7 QTL in this period, while in the recovery phase, yeah, we found 6 QTL, and only 1 QTL was shared, and that was this red QTL, which is already here and was coming up also in the drought phase, because, yeah, but this is already, let's say, prior the heavy drought stress phase. It disappears with strong drought and comes up again in the recovery phase, while the other clearly drought related QTL, they are not present in the recovery phase. So what we then did is that we compared and sorted all the QTL according to their appearance in the treatment and the timing. And this is basically the, the result of that. So each graph represents the log P values of a QTL or one marker from the QTL over time under well-watered conditions. This is black, and under the light gray circles, this is the drought stress experiments. And then you can identify in total 4 QTL, which play a role only under drought conditions in the late drought stress phase. So they don't come up under well-watered conditions at all, but they clearly come up under drought conditions. So there must be some drought tolerance genes in that region. And in the recovery phase, we found only 1 QTL, which was specifically coming up in the drought recovery phase and not appearing in the well-water treatment. And yeah, this was expected because here you had a low correlation of biomass of the 2 treatments, while here you had already a high correlation. So you wouldn't expect many drought-specific QTLs. However, 1 we could find. And the majority of QTLs, which are here 8 in total, we call constitutive biomass QTL because they were found in both of the studies, although still there are differences of importance. And of course, we looked also for the candidate genes, and yeah, let's say the highlights are here. So we have flowering time genes here in bluish. We have drought-related candidates here. And in green, I highlighted some general genes which are known to be involved in growth or early seedling vigor. And we can't really go into detail that. It's detailed discussed in the corresponding papers. But let's say Here we always suspect could be the flowering time gene be connected with this drought locus, but on the other hand, flowering time gene you would expect to have an influence on both well-watered then condition. If it has a general impact on biomass, you should detect them also under well-watered conditions, which we didn't. So it's probably more likely that it's really the drought-related candidate genes in that region. While here, for example, the other flowering time gene PPDH1, which definitely is proven to have an influence on biomass growth in barley, this is detected then in both of the treatments towards the heading stage. So that also totally makes sense. This was the most important biomass QTL under drought stress conditions, while in the well-watered treatment, it was this region here on 7H in the region of HV-DIM. That's a strong candidate. It's involved in the brasinosteroid synthesis, plant hormone regulating growth. And we are having resequencing data for this gene and look now on the candidate gene associations to that gene. We later also looked on how this drought period influences the yield parameters with a subset of the collection, so not suited for GWAS. So we took 26 contrasting lines, repeated the experiments, and went to maturity. And just very quickly, so this, let's say, early drought period caused a reduction in grain yield of 29%. Straw yield, not that much because the plants recovered a lot of tiller, but they were unproductive. And you'll see that here you have plus 19% in tiller number, but still the grain yield penalty is strong. So almost 30% less grain yield after such a pre-anthesis drought. And we looked a bit on contrasting genotypes, and we found here this landrace BBCH 1480 is a Russian landrace that has almost the same yield under control and drought conditions, so has a very low reduction, while another landrace has a very strong reduction under drought in the grain yield. So that could be contrasting genotypes for different grain yield under drought. So we have done this in wheat as well, but I think I will skip that slide. So this should be the last slide because we are advancing in the time. So as said before, we have not only looked non-invasively on the growth and well-watered and control and drought stress conditions. We have this panel was also subjected to root analysis in 3 different studies, which were all published last year, funnily. So this is a study with the CIGAR roll technique, where we looked on root and shoot growth in the seedling stage under well-watered, or let's say optimum conditions and osmotic stress. Then there was a study here by Nico von Viren's group who looked with the same collection on roots from rhizotron approaches. And there was another study from the University of Bonn, which had the collection under a rainout shelter in the field and did shovelomics at the heading stage. So, and this map is the QTL map from Abdelghani, and I try to highlight the overlapping QTL regions. So the 2 blue stars refer to the other root studies with the same panel. So it's the same barley panel, and you see that a lot of QTL regions are shared. There's of course more overlap between the 2 seedling stage approaches, but there's also overlap of certain QTL regions that were found by shovelomics in the field at heading stage. And a very interesting region is here on 6H that was exclusively associated with roots. And whereas a strong candidate gene is the PIN7, which is a pin-formed gene, which are known to encode auxine transporters, and they control radial root growth in barley, or not only in barley. And this was found by all 3 studies. And we had SNPs by this GBS approach from sequencing or fingerprinting the gene bank accessions. And there were SNPs from within the gene, and they associated even higher with our root traits than the iSelect marker from the genome-wide association scans. So it's a very strong candidate for this locus. But moreover, we also looked for co-localization of root traits and above ground biomass. And we do find also regions which we find in the, let's say, non-invasive phenotyping biomass studies that overlap with root QTLs here. So there's also somehow a cross-link on the genetic level there. This we will skip because the time is passing by too fast. And so I just want to summarize more or less this result. So first of all, we saw that growth per se, but also growth under abiotic stress is really highly polygenic and influenced by the environment. But with this controlled condition phenotyping and the non-invasive approach, you can really resolve the dynamics of the genetic architecture of these traits and also explore these trait relationships during the life cycle. You have the possibility to model the growth curves and reveal more traits which are not accessible otherwise. And you can do trait integration, and that reveals drought-related traits which are useful to classic genotypes according to this drought tolerance or pre-anthesis drought tolerance. And this, for example, this wilting time or growth stop that occurs at around 20% of water content. Also, the photosynthesis was shown to be decreased around that time, but when the drought ends early enough and not damages the plants, then you can recover also this very quickly once the plants receive water again. And the only implication on these diverse panels differing a lot in biomass is that this wilting time depends on the biomass at the onset of stress. And if you want to look at this trait in more detail, you need to establish mapping panels which have a similar starting biomass and also flowering time for a finer analysis. What we also saw is there might be more diversity in the land traces for drought tolerance, but yeah, this is another topic. And we saw, for example, that in the barley landrace panel or mainly landrace panel, the vegetative growth correlated with grain yield in both treatments, while this is not the case in this winter wheat panel, but this would be a complete different talk. And what we also saw is that this panel established from the IPK gene bank is really a useful resource for the exploration of many complex traits that are contributing to drought tolerance, and not only drought tolerance, it was also shown to be useful for salt tolerance, for example. Yeah, I know it was a lot of content. I hope you could follow, and I'm happy to take questions, but I first have to acknowledge, of course, you don't do such a study alone. There are many people involved that have helped with conducting the experiment, the analysis, and of course, big thanks goes to the quantitative genetics group. It was great collaborating with them on analyzing these data sets. And of course, on the image analysis group with a former member, Christian Klukas, Dijon, Jean-Michel, Ingo, and of course also the recent members, because we still need to do image analysis, and we try to also develop algorithms which tackle some more critical traits. Big thanks to Adel Abdelghani for conducting this root study, and all the other colleagues which were involved. And a big thanks to the audience. Although I cannot see you, I hope you could follow, and now I'm happy to take questions.
Thank you, Dr. Kerstin. A very well designed, excellent presentation depicting excellent work carried out by you. As researchers, everyone has a doubt how the genes send those signals and how those things are expressed. We all have studied gene expression in our college times, but seeing those signals being translated into different characters, and those characters expressing at those particular stages of time and capturing those things very precisely at those particular times is very, very important. And that is the beauty of the phenomics which you have shown in your presentation. And also she has shown how the biomass is being varying with time to time before and after the stress, and how the various drought QTLs are playing their role at different times. And she has made a beautiful connection between the phenotype, the genotype, and the technology which was used in capturing these traits? I think Dr. Raju, there are many questions. You can just put the question.
Yes. Thank you very much, Tara madam, and thank you very much, Kerstin, for great presentation. You have been watched by more than 1,600 people, and I am having several questions. Okay, can we request all other colleagues to mute the mic, please? Prasad, can you mute the mic? Zoom some questions from that YouTube, and Christine, you can respond some of these things here, and at later stage, as I told to all the participants, they can talk to us through Twitter or Facebook. We can get those answers again. So now the first, well, there are many questions, not in order. You can answer. Duration and intensity of drought stress influence growth and yield of plant. In your opinion, which could be given more importance for phenotyping, duration or intensity?
That's a good question. So I mean, drought is an easy word, but it's a very complex term because, of course, it depends a lot on the environmental factors, and there are a lot of things that you maybe cannot model in a pot experiment. So it's a particular type of drought which you can model there. But of course, intensity is how long the drought occurs is a very relevant factor, but all the other environmental factors as well. How is the soil type? Is it a light soil, like a sandy soil we have here in the northern German area? Or is it a heavy soil that can hold the water for a long time, so plants will dry out or the soil will dry out slower? And
Okay.
what is the temperature at that time? So we can model certain things. I mean, we could explore different soil in this system. We can, with the, let's say, technique that the greenhouse allows, also conduct heat stress experiments. We have done this already, and we're at the moment exploring on 2 genotypes in a pre-experiment, heat and drought, and the combination of heat and drought on winter wheat, high-yielding cultivars. So
Good.
you could make 1,000 drought scenarios, but you need to decide for one and let's say aim at the scenario which is maybe in your region, more often occurring.
Good. Other question is temperature fluctuation more harmful or stable hot temperature? So which in your opinion is more harmful for the yield for the crop? So temperature fluctuation or stable hot temperature?
That's that's a question we haven't addressed.
Okay.
So in general, a high temperature has all. I mean, it's similar with drought. When does it hit the plant? So is it in the grain filling stage? It's maybe not so painful as in the flowering time stage where the pollen is affected and so on. Nothing or too little to nothing is known about heat effects on the early growth stages, because here they don't really appear. So you have heat mainly in the grain filling stages or around the flowering stage, but in the early growth stage phase, that's not really investigated. However, that might change with climate change as well. So we see that the hot days get earlier and earlier. But I think what's really very severely affecting yield is when you have a longer heat wave. So plants cannot breathe in between, let's say, that temperatures goes up and down. So I would say the long-term heat has a higher effect.
And many people, majority of the people appreciated your presentation. They really liked the presentation, your slides, and many of them, they are congratulating you for the huge work which you have done. Some people have asked some technical questions, that which program, which software you use. Some people asked, what is the cost of establishing those kind of phenotyping platform? So I don't think that we need to do. But
that's a talk on its own.
A few other questions. How to phenotype only heat tolerance? So they are talking only heat tolerance in respect of biomass. How to standardize stress frequency and the duration of stress? So because sometimes this temperature and drought, they are together kind of thing. How you measure?
Yeah, this you can, of course, nicely do in the greenhouse or in this plant cultivation hall, because you have full control over when the heat should appear. So you switch on the heating or not. So in the field, it's unpredictable. And you have the advantage that you can water the plants. So because we do daily watering and keep them at a very high water content, they do not run into a drought. So because we have an ongoing experiment on heat tolerance right now, and we have subjected plants to a really strong heat of 35 degrees. So we took plants from the LemnaTech to the heat stressed climate chamber, and then we continued watering daily. And the water content from one day to another goes from 90 to 50%. So that's the point. You have to avoid that the plants run into drought stress during the heat, and then you have the effect of heat only. So that's doable. Probably not in the field, but in such systems, yes.
Yeah. Phenotyping may be influenced by limited soil fertility and diseases. How can these be addressed during the phenotyping?
So we are controlling the diseases. I mean, we're a greenhouse that the windows are sometimes open, so you can have powdery mildew, you can have aphids, for example. So if we discover symptoms like this on few plants, we spray. So we control this because we do not want to have a mix of disease and abiotic stress.
Yeah.
And of course, soil fertility, you have to decide for the soil and test this substrate and apply fertilizer if you go over a longer period of time. So if you don't look just in an early growth stage, you need for sure to fertilize the plants. Yeah. This needs all calibration. That takes a lot of time.
Yeah.
Now.
And what is that correlation with drought and photosynthesis activity during and after wilting process, as your biomass decreased and activity increased before giving water?
Yeah, basically that's a tricky question because you have to imagine we had this setup where we rewatered the plants. So the photosynthesis was mainly affected in the drought stress phase. And if that still correlates with later yield, this we are seeing at the moment, and we have done this because this was installed later. This was first implemented in 2017. That was during the VII-NGGIBCI project where we evaluated the spring wheat collection. And Michele Greco was here. He's a photosynthesis expert. He has developed these measuring protocols for this chlorophyll fluorescence camera system. And he explored these traits. And this is a manuscript where he wants to send me the draft this week. So we have exome capture data for this. But we saw co-localization of photosynthesis QTL and biomass QTL, that's for sure. And under stress, but I have to see the draft of the paper first, he also said there is a connection with the grain yield. It's not a very high connection though, but it's absent under well-watered conditions.
Other quick thing is, what is the relation of plant microbiome to the phenotypes of the crops in abiotic stresses?
Yeah, I mean, we cannot evaluate that. There are some studies on mycorrhiza, for example, also by the JKI in Quedlinburg by Lehnert et al. 2017, who looked in wheat on mycorrhiza, and they could show that first of all there is genetic difference of how good the colonization is, but also that it can really improve the grain yield under drought conditions.
Okay, maybe last question, although there are many questions, but no problem. Last question is, how effective imaging-based phenotyping over conventional destructive phenotyping activities? Any comparative percentage change or efficiency to appreciate new imaging-based studies? You got the point?
Not fully. So do you mean field versus spot comparison? Or?
Yeah, so basically when you are having this digital or this image-based phenotyping versus conventional, and then in terms of the cost or in terms of the efficiency, so have you compared those kind of things, something like that?
Oh man, I'm not an economist, but I mean, these phenotyping systems, I know every big breeding company is having such systems. So there must be a kind of value for them to invest in these high costs. And they also all work with drones in the field. It's still the question, can you assess the same traits from this, let's say, daily imaging and really controlled phenotyping, while in the field you maybe cannot always acquire the images that you want because there's too much wind or the weather isn't right. So you have, I would always go for such traits. You first conduct such an experiment in very controlled conditions and then try to translate it to the field, because you know already at what periods you should look and how. But it's of course not the same. You can make more detailed analysis in these very controlled conditions.
Very good, Kerstin. I know there are a lot of questions. So as I said, so all these colleagues who are having good questions, other questions, they can contact us, the organizers. And I already mentioned our Twitter handle and Facebook thing. So you please follow there. We will get you those answers. Sorry, we cannot take many more questions. But again, great presentation, Kerstin, from you. I know that you were having relaxed time during lockdown period, but because of me, you had to prepare, you had to work hard. And
we did our best to assemble a short.
Yeah, so thank you very much for doing that. Now I give the floor back to Tara, madam, again. And thank you once again, Christina.